This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1161, recorded on October 18, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And here in Western Mass, it is another cloudless, glorious autumn New England day with the leaves changing. We're at about peak foliage here at 68 Fahrenheit, 20 C. And we have had no rain in ages.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. My weather is almost exactly like Alan's. 67, but otherwise just like that, though I'm not sure we're at peak foliage yet.
You're probably about a week away.
Yeah.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Well, in the continuing trend of temperatures, I'm 66 Fahrenheit, which is 17 or 18 Celsius, and it's sunny and beautiful, and I'm not sure about peak foliage either, but close.
All right, now we're going to go in the other direction. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Great to see everybody, actually. The weather in Austin has been and continues to be weird. We last week had a record latest day in the year of 100 degrees. Okay, that was just a few days ago, where we hit 102, and that is 11 days past the previous record, and this is since records have been being kept. So, welcome climate change. But right now, then we had a cold front come through, and the temperatures dropped 25 degrees. Right now, it is 79 and overcast. So it's kind of at least it's not stinking hot. I'm not used to any clouds in the sky, so that's a bit shocking. And we have the beginnings of what passes for fall in Austin. I think some of the leaves are getting tired. They don't really turn. The fall asters are blooming. That's the good news. They're really pretty.
Oh, nice.
If you like these programs, these science programs, please consider supporting them. Microbe TV is a nonprofit in the U.S., and so your donations would be U.S. federal tax deductible. You can go to microbe.tv/contribute for various ways that you can contribute and help us out. We're coming up on our fundraising season, November, December 2024. So it's your chance.
Microbe TV has a fundraising season?
Yeah, now we're going to have a fundraising season.
Oh, okay.
We need to get serious about this.
Well, end of year is everybody's fundraising season, right?
Can we have events and stuff?
What kind of events would you like
to have?
I
don't know. I'll think of something.
Well,
we'll get back to our regular programming after we get 5 more callers.
Yeah, there you go.
Yeah, we're going to have some things. We're going to give stuff away. Yeah. We're going to give away. People can donate to listen in live on a TWiV like this, right? We'll give them a link. They can have dinner with us, and stuff,
Yeah.
That reminds me, I met all kinds of people at TWiV 1000 who were from Ann Arbor, and I said, well, let's do lunch.
Yeah.
And I haven't heard from any of them, so they know where I am if they're still listening.
There are a few Austin TWiV listeners, and I have had lunch with one or two of them on occasion, but there's a lot more potential out there.
I've heard from, over the years, a couple of people who are in Western Mass who listen and we've never gotten together.
Anyway, keep us in mind when you are giving away your funds this year. We'd love to have your help. A few news items for you. We have a polio report from a website called reliefweb.int, and new isolates continue to be found throughout the world. We're now up to 55 countries globally that have reported poliovirus isolates since the 2016 switch, removing type 2 from the vaccine. I made a figure this week showing cases of polio before and after the switch. Oh my gosh, a few hundred before the switch caused by type 2 and over 3,300 after the switch caused by circulating vaccine-derived type 2. Anyway, now this week we have isolation of CVDPV2 from Spain, from Barcelona, where it's never been isolated before. Some from some type 1 cases, wild type 1 in particular, from Pakistan, circulating type 2 from Angola, French Guiana, Niger, Nigeria, South Sudan. I mean, basically in Africa, type 2 circulating vaccine-derived poliovirus, a human-made virus, is endemic. Just want you to think about that, folks.
This idea of pulling the serotypes out of the oral vaccine, it feels almost like a Jenga game that's gone horribly wrong. Right?
It is.
Just like, we'll just pull this out. Here we are.
Yeah, the architect of that plan, Roland Sutter, has written an article saying this was a disaster. It was a bad move. And it was all based on the computer modeling, which said if you took the type 2 component out, the type 2 circulation would gradually go to zero. I wonder what data they put in for that.
And wasn't there some work by these people? I don't know if you've heard of them, Racaniello and Dove.
No, never.
that seems to talk about this at the time. That thought it was a bad idea.
They don't know what they're talking about.
They were uninformed.
Yeah.
We're always uninformed. We in the 90s, we wrote an opinion in Science, Alan and I, saying, this is a problem to think you're going to stop vaccinating. And, little did we know they were going to stop vaccinating against type 2, right?
We didn't anticipate this particular move, but yeah.
So is there a movement afoot? I mean,
No.
What's going to be done?
So I and others have been petitioning to change the vaccine. And one of the more egregious things is by June 2025, nobody is supposed to be working on polio anymore. You have to destroy all your stocks except in what's called a polio essential facility. You have to be approved nationally to do that, and it's expensive, so most places are not going to do it. And the logic is we're so close to eliminating polio, there's more polio in labs than there is in the environment, so let's get rid of it in the labs. Well, that logic is flawed. There's more in the environment. So we have this one case in Spain. That means there's at least 100 people infected around them. So, it wasn't a case, sorry, but you get the point. Yeah, this is a big problem. And unfortunately, the program is being run by bureaucrats who have no science experience. They just are numbers people who say, yeah, we can do this, but they don't know any science. So we wrote letters to people who are in charge of this containment. And they say, no, it's going to go as planned. We're not changing.
Doesn't seem to be going as planned.
No.
And there are more and more cases. There's more and more circulation. I think clearly the thing to do at this point is to switch to IPV. But
switch to IPV, go back to trivalent if you're going to be using OPV, because obviously type 2 is circulating again, thanks to this move. And just resign ourselves to the fact that until we can give everybody IPV, we're going to have to keep vaccinating.
There's no plan to switch to trivalent. And although some, as you know, type 2 was modified to be NOPV2.
Right. And
they've also modified 1 and 3. And so they want to make a trivalent out of that, which I don't think is an improvement because NOPV2 is really marginally an improvement. But in most of the world, they use bivalent still. So Pakistan, Afghanistan, where you have gangbusters type 1 circulation, they use bivalent still. And they get cases of type 2 as well because there's no type 2 immunity. Anyway, I don't want to spend all the time on this, but it's a big problem. And also I noticed this week, I used to go to polioeradication.org. They had lovely maps every week with all the new polio cases. They took it down because they don't want to show it to us anymore. Because it's embarrassing.
Yeah.
So what do you do when you don't like the data? You just
You just
sweep it under the rug.
shuffle it away.
Yeah,
of course.
Polio is to the World Health Organization what Fidel Castro was to the CIA. It's just proudly eradicating it for 50 years. And
I mean, if you're doing something for that many years and it's not working, don't you think you need to change your plan?
yeah.
Seems like it.
What's
Anyway.
that quote? The definition of insanity is
Yeah.
doing the same thing over and over again and expecting a different result.
Which is
It
not
is.
an actual definition of insanity, by the way. But
So I go to Relief Web now to get some information. They don't have maps, but it's not nice, polioeradication.org. But polioeradication.org is run by the Global Polio Eradication Initiative. And of course they're going to take off the data. I have to show you the map I made sometime. It's really impressive. Okay. Another piece of
the
news.
next news item is actually about pulling something out of a vaccine, and it's probably a good idea this time.
Yeah,
I
this
know.
is what we would love to have things look like.
Yeah. So it turns out I pasted this in here, and I had seen discussion of it before, but this is a nice NPR article that summarizes what's going on. The B Yamagata lineage of flu has, as far as anybody's been able to tell, gone extinct. Nobody can find it. And this happened during the COVID-19 pandemic. So there are a couple of influenza B lineages that have been around for a long, long time, and they are part of the quadrivalent vaccine. B Yamagata during the pandemic was at low levels, and then the levels got lower, and then it disappeared. And so what seems to have happened is that all of the isolation and masking and such that was trying to control SARS-CoV-2 successfully controlled a strain of flu and wiped it out. And so this year's flu shot in the Northern Hemisphere does not have B Yamagata in it. It's not there. And the reason they did that was because they realized, the only place you find this is in vaccine manufacturing facilities where they have vast quantities of it growing in eggs, and somebody could get exposed and reintroduce it. So why don't we take advantage of this opportunity? And in the case of flu vaccine, there's a new one every year. So if it does come back, we can just put this strain back in there. This is not, and it's fairly low risk to remove it at this point. But I thought that was a really neat piece of news.
So a little message to the anti-vaxxers out there. People do have an ongoing risk-benefit analysis for vaccines, which is what this is. Although the risk is minuscule, the benefit at this point from including this in the vaccine is also minuscule. So it's not even worth the minuscule risk, not to mention that if you take it out, it makes the manufacturing easier, which I think you already spoke about. So, the people who understand that vaccination is a good idea also understand that there are circumstances under which it's appropriate to remove the vaccines. There you go.
Yeah.
It's all rational.
Personal experience for me, before I went to Australia, I got the new COVID-19 and the 2024-2025 flu. And I was very insistent. This is going to be the high dose quadrivalent, right? They said, yes, high dose quadrivalent. And as he was giving it to me, I said, this is high dose quadrivalent, right? He said, yes. And then when I got the printout, it was flu zone high dose trivalent. And so then I was like, wait, what happened? And so I didn't exactly know that it was because they took out the B Yamagata, but then it came to pass that
That's it.
people
the
were
quadrivalent
informed.
is no longer a thing.
Right.
All I can say is I hope they're right.
Yeah.
Yes. But as I said, since flu shots come around every year, if this turns out to have been premature and Yamagata comes back, put it back in next year.
Yeah.
Yeah. Well, it's also kind of interesting because I've been hearing bits and pieces about Yamagata maybe being extinct for a while, and I don't know that we had expected it. And so it makes us sort of think what was going on differently about this virus and how come it became extinct. And what was going on with it in the reservoir. And it also shows us, I think, how much we still have to learn, even though we know all sorts of excellent and fancy stuff that we're going to talk about in future papers.
Yes. Yeah. And this was wiped out apparently by old tech, like ancient, just isolation and masks. And this virus, flu, is a little less contagious than SARS-CoV-2. And that was apparently enough of a difference to eliminate an entire strain.
And finally, this week were the Nobel Prizes announced. And I just want to note one of them, Physiology or Medicine, to Victor Ambros and Gary Ruvkun. Victor's at UMass Medical School, and Ruvkun is at Harvard and Mass General Hospital for discovering microRNAs and their role in gene regulation. And I just want to say that Victor Ambros was a graduate student with David Baltimore at MIT, and I arrived there. He had finished and had moved on to a postdoc with Robert Horvitz, where he was learning C. elegans, and then he started his own lab on C. elegans, and that's where he discovered microRNA regulation. But he came over to see me several times and talk about polio, and we got to be good friends. And his wife was also working there, and I taught her how to sequence. We're using these huge gels, Cindy. I think her name was Cindy. No, I forgot her name.
She's actually referenced in this article.
Yes, it says Rosalind Candy Lee.
Yeah, Candy. I used to call her Candy, and I taught her how to sequence. And then she got pregnant at one point, and she was like 8 months, and she insisted on handling these big plates by herself, because I would say, let me carry that. And I said, no. So anyway, then he made a little company to make gel boxes, right, to run gels on, because companies weren't making them anymore. So he made his own company and making. When I moved to Columbia and opened my lab, I bought a bunch of them. So here he wins a Nobel Prize.
Nice.
I wonder if he has the same company. Anyway, then one of my students went on to do a postdoc with him, and it's cool that this happened. It's very
This
nice.
article I really liked. It stresses the role of serendipity,
Yes.
in important discoveries, and it also stresses the role of what to the outside world look like weirdo model organisms. Okay.
Yeah, it stresses
So-
basic research.
Yeah, this is very basic research, and I think that the article is really absolutely, it's beautifully written, and I think quite understandable to the lay public, and really makes those points really well. There's this quote in it from one of them that is like what we've talked about before, about where they, as they were doing this stuff, they weren't thinking, oh, we're going to win a Nobel Prize. They were thinking, oh, this is interesting.
This is weird.
I wonder if they all do that.
Yeah. Good job by Catherine Offord on the.
So, by the way, this is the fifth RNA Nobel for RNA.
Mm-hmm.
I think this is cool. mRNA got a Nobel Prize in '65. Discovery of mRNA. The enzymatic activity of RNA was discovered, and it was awarded in 1989. RNA silencing in 2006, and then this year's and last year's.
So I have
RNA
a bone
interference.
to pick with microRNA,
Uh-huh.
or with the creator, if you like. Okay, because it's kind of, we made this messenger RNA. Oh, maybe we shouldn't have that now. Let's get rid of it. Okay, why not go back and just not make the messenger RNA? I guess that's too complicated.
It is so bureaucratic and so typical of evolutionary processes, isn't it?
Thank you, Alan. I have often described I call it the molecular bureaucracy of the cell.
Yes.
Okay. And in particular, I haven't done this for a while. I had this conversation I had with a bike riding companion at one point, where he posited he didn't, he was a finance guy. He wasn't a biology type. He posited that, after 4 billion years of evolution, the genome must run like a finely tuned engine, and everything is there for a reason and stuff. And I said, that's when I came up with the bureaucracy idea. I said, he was a professor. I said, you're familiar with university bureaucracy. If somebody's not doing their job, if you got a process, there's somebody in the middle who's not doing their job, do you fire them? No, you hire somebody else and go around them. Okay, and that's exactly the way evolution works. So you wind up with this giant bureaucracy with a bunch of stuff that doesn't work quite right and a bunch of bug fixes.
It's so Parkinson's law applies even at the microbial level. Yes.
Yeah. So I gotta. I'm sure that Victor and Gary are listening. So I want them to tell us about this. Are microRNAs just a bug fix, or do you have a better explanation for this?
I mean, it does seem like they give us more options for control and more options for customization.
Well,
and there's a lot of genome devoted to this, right?
Yeah.
Look, there's no reason why they couldn't have been there at the onset of mRNAs already, right?
No, there's a thought.
I don't see that spot. That we're assuming they came sequentially, but evolution's not about the future; it's about now. Right. So sometimes you make stuff, and as you say, it doesn't work in the future. But I suspect the microRNAs were around doing things before we had protein coding
from
mRNAs,
the get-go.
Right?
Interesting.
Maybe. Okay, on to the literature, and today our snippet is from Kathy.
So this is a paper entitled RAAV, which is recombinant adeno-associated virus, expressing a COBRA-designed influenza hemagglutinin generates a protective and durable adaptive immune response with a single dose. So a couple things in here that we'll explain in more detail, but I wanted to tell you that I talked to the two first authors, who are Kristen Wiggins and Stephen Winston, co-first authors. The additional authors are Reeves, Givert, Spence, Brimble, Livingston, Morton, Thomas, Sant, Ted Ross, Andrew Davidoff, and Stacy Solch-Cherry. And I think Kristen is a student of Stacy's, and Stephen is a just recently graduated student of Andrew Davidoff's. And so Stephen and Andrew are the AAV component of this, and Stacy and Kristen are the flu component. And so...
and they are at, so this crew is at St. Jude, and also University of Rochester, Cleveland Clinic, and Cleveland Clinic.
Yeah, Paul Thomas has moved from Georgia to the Cleveland Clinic in Florida. So he's the one who's the contributor of the COBRA technology that we'll tell you about a little more detail also as we go through. But I would just want to give you the summary. The title kind of gives you the summary, but the idea is, of course, so we would like better influenza vaccines. And having just come back from this big Options 12, it's called Options for the Control of Influenza. It meets every 2 years. Also this year had coronavirus and respiratory syncytial virus, but it's predominantly influenza. This idea of better vaccines is a major discussion of this. And so I was looking back into Ted Ross's background, and he wrote some article that was from about 10 years ago that said there were at least 40 universal vaccine projects in progress, and that was 10 years ago. So you can just imagine what it's like now. And we heard all kinds of different things at this meeting in Australia. But we didn't hear this particular work that is in this paper today. So there's a lot of different approaches for these universal vaccines. They can be to the influenza stalk or other parts of the hemagglutinin or other proteins, for instance, the M2 protein of flu. And so this paper uses this technique from Ted Ross, which is to bioinformatically design a broadly reactive antigen. In this case, it's the hemagglutinin, which we also know as HA or H. But it's also been done for neuraminidase, which we also know as NA or N. And so
did they use all HAs or just H1?
I think in this one it's just H1.
Okay. That's what I thought.
But I was in the weeds with the Cobra and all the different ones that they used, and
when you're in the weeds with a Cobra, then things get
Yeah, you got to be careful.
right.
You
Yes.
explain. I'm sorry, I had to step away for a second. You explained the Cobra abbreviation.
No, I'm just about to do that.
Okay,
It's
sorry.
computationally optimized broadly reactive antigen. Okay.
It says it generated from a consensus of H1 strains.
Okay.
Okay.
So
Which is still pretty good. I mean, if we just got rid of H1, that'd be great. We're pretty excited about Yamagata.
Yeah.
minutes ago.
right. Right. So the idea is to design this broadly reactive antigen, and before I get into the details of how they do that, it was known that making these broadly reactive antigens as a soluble protein and using those to immunize was protective and efficacious and so forth. But that was just using it as a soluble protein. So what this paper adds is taking that kind of Cobra bioinformationally, computationally designed antigen and putting it into an adeno-associated virus vector. And so we'll talk a little more about that too. And so the other tweaks that they do is that they modified the antigen to make it more immunogenic, and so they call that self-adjuvanting. So it kind of is just a one-stop thing. And they found that they could just give it once at a relatively low dose intramuscularly and protect mice from a challenge. And then they did a very preliminary pilot experiment at the end with ferrets and got positive results with that too. So basically, it's this proof of concept that they have these vectors, adeno-associated vectors, these computationally designed antigens, and you could do this with lots of viruses and lots of different antigens. So that's the summary.
I think proof of concept is an important phrase here.
Right.
They're not rushing into phase 1 trials or anything like that.
Right.
There are, as we will see, there are issues that need to be addressed, but they're the kinds of things where you don't want to waste your time trying to address those issues if it's not going to work at all. Okay.
Right.
So let's see if it's going to work at all.
Right. So adeno-associated virus is a single-strand DNA virus, very small. It's in the parvovirus group. As I like to say in class, parvo means small in Latin. And so these are small.
Wow. I didn't know that.
Yeah, parvis is the Latin word for small. Yeah. So anyway, so they're very small. So what they've done in this is they've taken out what small amount of important stuff they need for the virus and put in this antigen that has control sequences and things to get it expressed well and so forth. And then on the end, it has the inverted terminal repeats. So if I couldn't figure out whether this is open access. I suspect
It is not.
maybe not. But 6 months after publication, which was in July, it will be open access. That's the ASM thing, I'm pretty sure. So not too much longer. Anyway, figure 1, they show the diagram of what the vector looks like.
And so the idea is that the cell who receives the vector, so the vaccinated cell is going to make the antigen. And so in some ways it's kind of like, mRNA vaccine or something that we're trying to make our cells make the antigen.
Yes. But in fact, what they actually end up administering to the mice is vectors. Virus vectors. So they have this in them instead of their genome. So it's this bit of DNA.
Right, but it's still, when it gets into a cell, will result in production of this protein by the cell.
Exactly. Right. And the adeno-associated part of it is that it's long been known that these viruses, which are also known as dependoviruses, depend on some activities from adenoviruses or some other things to help them grow. And so
and these adeno-associated viruses are a long-standing choice for gene therapy because you can gut them and get the virus out, and because they're very, very safe. It can't even complete its infectious cycle without an adenovirus there to help it, and you've gutted it. So this is like the most innocuous virus you could get.
Right. And so this brings up a point that when I was talking to Kristen and Stephen, that came up about adeno-associated viruses, is that because they are so extensively used for gene therapy, they've done all kinds of things to make them non-immunogenic. So there's all kinds of knowledge there. And that now when you're trying to use them as vaccines, you can take that knowledge and like put those things back into the virus. And that'll come up a little later. And the same is true for adenoviruses. All the kinds of things that people have been doing to make adenoviruses gene therapy vectors, people reverse when they're trying to make adenoviruses into vaccine vectors. So, so let's see, where was I? Yeah. So to make this HA antigen, they get a bunch of nucleotide sequences, in this case from H1 viruses, and they get it from a group of viruses. The nucleotide sequences, they translate those in silico, and then they do alignments of those, and they try and figure out what's the best consensus. And then they can do that iteratively. They could add in different bunches of sequences from influenzas that have been sequenced from different sites, or all different kinds of things. That's kind of the weeds I got lost in the Ted Ross papers. But anyway, so they use these collections, and so they call it this layered approach. But Stephen used the word iterative, which I like.
So, I mean, my sort of rudimentary understanding was that of all these different H1s around, you take, in terms of immunization, what you think are the best bits of all of them and stick them together, or make some sort of consensus.
This is
and
the
best
H1
bits
highlight
would
reel.
be not only the most immunogenic, but broadly immunogenic against a bunch of different versions.
You're seeing
but you
it
end up with one sequence that you put in the vector, right?
Right.
Mm-hmm.
Yeah.
Right. But it's representative of presumably different geographical areas, maybe even temporally, over a time span and so forth.
It's chimeric in the classical sense, where it's got like the head of a chicken and the body of a dragon type of thing. So you've got pieces of all these H1s represented on this single antigen.
I can imagine that in the long run, this would be a place for AI. Hey,
Sure.
here's all of these different H1s, and here's their different properties. Make me an H1 that combines the best of all of these for a vaccine.
So what I don't get is, let's say you have an antigenic site, a known antigenic site in one isolate, and we know antibodies are directed against that. And then another isolate, there's a one amino acid change that abrogates binding. So which sequence do you put in? One or the other?
Well, I think the idea is you assume that the one that abrogates binding has a nice binding site elsewhere in the protein.
Yeah, I know, but maybe the other one is still around, so.
Yeah.
I also wondered as I watched this, I looked to see if anybody had ever, made this protein and done a structure. Okay.
Yeah, it's interesting.
Presumably it would fold like a regular hemagglutinin, but who knows?
Well, given the results, I guess it does.
Yeah, apparently.
Yeah. Again, when I was in the weeds with those papers, I have the feeling I saw something looking at things structurally, but I can't swear about that.
Well, and that's also a nice part of this being in AAV. Before, Kathy mentioned that they've done it soluble, and that isn't going to necessarily give you the perfect structure because it's usually a membrane-bound protein. But when you're having it made with AAV, you're getting it in that correct structure.
I bet they used something like AlphaFold.
Yeah, right.
Which also got a Nobel this week, right?
Right.
Good point. You don't have to determine a structure anymore.
Right. So I did ask Kristen and Stephen, this is taking a cue from Michelle Swanson, who does this on the This Week in Microbiology. When she talks to authors, I said, what was the most exciting or surprising or yippee moment in the whole process of this work? And they said, well, actually, it was probably the experiments in Figure 1. That was like the shot on goal, and we didn't expect it to work quite so well. So now I'll tell you what they did in Figure 1. Basically, they took this vector And it was with a particular strain of AAV called AAV8, because that's what the Davidoff lab uses a lot for a variety of reasons. And they put in this optimized hemagglutinin, the COBRA optimized sequence, and they created the virus stocks and used 10 to the 10th, 10 to the 11th, and 10 to the 12th. Their unit is called vector genomes. And they also used controls that instead of the antigen, the HA antigen that had a GFP, or they used a control that was just PBS. There was no vector whatsoever, I guess. Okay. So they stuck those into mice and then intramuscularly. And then 6 weeks later, they challenged them with a lethal dose of an influenza strain called California 09. And that's an H1N1 virus. And not surprisingly, all the control mice lost weight and died. But all three of the experimental groups, no matter what dose they got, they all survived. They were protected from weight loss, and they were protected from death. And they even looked, before they challenged them with the California 09 virus, for hemagglutinin inhibition activity, HAI activity, which is sort of the gold standard assay for humoral response, B-cell response. Its correlative protection for the antibody response is this HAI assay that all the influenza people do.
So it's interesting that when you talk to them, they said this experiment was the exciting thing, because you're reading a paper, okay? Obviously, it's been published, so stuff worked, okay? You're reading the paper, and you look at figure 1 and you say, oh, yeah, not only does it work, but it works pretty well, because even the low dose gave you complete infection. But it's sort of, yeah, okay, check that box, move on. But if you're doing the experiments, you don't know.
Right.
It might be a complete flop.
Yeah.
Okay, right. So the aha moment there is, ooh, great, we've got a project.
Right. Yes, I agree. Okay. So then, as I said, they used AAV8 in this, but they know that there's lots of different AAVs, and so they screened a bunch of them and figured out that AAV9 was the one that they wanted to go forward with for a couple of reasons. It gave a good preliminary HAI activity. It had the least weight loss. It had the best production of virus stocks when they did their preparations. And it was predicted to have less hepatic toxicity. And hepatic toxicity can be a problem with AAVs. And so that was why they chose to go forward with that. So they called this new vector from the AA9 V2. So nicely throughout this paper, they just have simple names for the viruses that the vectors that they use. V1 was the AAV8 one that they used in figure 1. And then V2 is the one that we're going to hear about next for a while. And then there's
version 2. Yes.
Version 2. Yeah, vector 2. Okay.
I want to take a minute here to just detail a little more about the vector technology. You've kind of said this, but I want to emphasize that in terms of the nucleic acid genome that is packaged in the virus, the only thing left of AAV is these non-coding sequences at the end that are necessary for the packaging of this thing in the virion, and also have the effect of down the road when it gets into the cell, causing this DNA to persist as an independent element in the nucleus for some period of time, which is something that I want to come back to at the end of this. But you don't have to, and that thing, as I understand it, can be packaged into any number of AAV capsids. And there is a whole zoo of AAV capsids that target different tissues and all of this kind of stuff that people have been tinkering with for a long time, all the way down to custom AAV capsids. Take what you know about these guys and try and design your own to target it in a certain way. But importantly, what you do is you take this genome and you introduce it into cells that are making the capsid protein. That gives you a preparation of virus, in air quotes, okay? That is this genome packaged into this protein shell that allows you to infect cells with this thing, hopefully in a targeted fashion. But after that, all you got is the DNA. The capsid goes away. You're done with that. You're not going to make any new stuff. You're not going to make any new virus products. You just have this persisting piece of DNA that's making these proteins. Fair enough?
Right.
Yep.
Good. Thanks. Yes. So the next thing they did then was test this new vector 2 against the standard of care, one of the quadrivalent flu vaccines called Flucelvax, which I thought was going to be a cellularly produced vaccine, but I had to double check it. I looked it up, and yes, it is produced in cells. It's an inactivated quadrivalent vaccine. So they tested mice with the Flucelvax and with this vector 2. And this experiment was in male mice, and they showed again that it was very effective at decreasing morbidity and mortality. And basically, this vector 2 was better than the Flucelvax in pretty much every assay that they compared them head to head. Okay, so that was in male mice. And you might ask, well, why do you do male mice? Well, I know that the reason we started doing them was A, because that's what everybody else had done for the virus we work on, and B, because they're cheaper than female mice. Okay, so I don't know if that was the reason why they did the male mice first, but it's known that there are sex-specific differences, particularly for influenza. And you only have to go back to our TWiV 624 with Sabra Klein, Sex Matters, to remember that that's the case for influenza in particular, that females can have more pathogenesis. And so they wanted to do this test in female mice. And then there were also some aspects.
Right. You don't want a vaccine that's only against the man flu.
Right. Right. And there's also some differences in the recombinant AAV in terms of liver toxicity that are both some sex-specific differences and some species-specific differences. So they tested this same V2 now in female mice, and again, they got good protection from their challenge virus. So it seemed to behave pretty much the same in the male and female mice. So let's see. Oh, yeah. So then they wanted to know whether these could be useful against animals, and then in your mind you're thinking, or people who had some previous antibodies to influenza due to exposure or vaccine or something like that. So they used the mice from that first experiment with vector 2, the one that they did in male mice, except that now, and they were collecting blood and maybe other, but anyway, samples all along. And then at 21 weeks, they had two groups of mice, those that had not gotten challenged and those that had gotten challenged. The ones that had gotten challenged originally, they now challenged with the California 09. And the ones that had gotten challenged with the California 09, they challenged with a heterologous virus, one called Hawaii 2019. Okay. And so then that's a drift variant of the California 09. So then they looked for protection, and in both groups, whether they had been previously immunized or previously challenged or not, there was good protection. And in those that got the heterologous challenge, now with the Hawaii 2019, the results were similar in terms of long-term protection to what they saw with the Flucelvax, the standard of care. But they did see that their HAI titers were better in the V2 vaccinated group, again pointing to maybe their vector being a little bit better than the standard of care. So next, they wanted to try improving the vector, but the situation was that they had such good protection at the doses they were given that they needed to have conditions where maybe the vector was a little bit less good, so that then they could try to see whether they had improved it. So they did what they called a dose de-escalation challenge study, so that any improvements that they might introduce would be discernible. And so they gave a variety of lower doses to see what was effective and what wasn't. So before, remember, they had given 10 to the 10th. Now in this de-escalation, they gave 10 to the 9th, 8th, 7th, and 6th. And they only saw protection in the 10 to the 9th, and lower than that, they didn't have good protection from the challenge. So they knew that they had to use a dose somewhere around 10 to the 9th. And so what they did then was try one way to make their antigen better based on some of the knowledge that they had from the gene therapy field with AAV. And that was to increase the number of CpGs. And so CpG dinucleotide pairs, if you have those, they will increase the immunogenicity. At least that's what was known from the gene therapy vector field for adeno-associated virus. So
yes, they bind to a toll-like receptor, TLR9,
right.
And induce an innate immune response, which helps make the adaptive immune response better.
Thank you. Yes. So their starting vector V2, they called low CpG, and then they increased the number of CpGs to 50% of the possible total, and that's called V3, and then to 100% of the possible CpGs is V4, and they called that high. So then they had these V2, V3, and V4, and they vaccinated mice now not with 10 to the 9th, but actually half of that, 5 times 10 to the 8th. So you would expect that if it was not, that the V2, based on their previous results, some of the V2 recipient mice wouldn't survive. But if you had improved it by including these CpGs, maybe the V3 and the V4 injected mice would do better. And in fact, that was what happened. The CpG enriched V3 and V4 injected mice were protected at this lower dose.
So I have a question here that clarification that maybe Brianne can help me out with, or anybody, but it may be a question for Brianne. The CpG, its activity is as a DNA dinucleotide. Is that correct?
Yes, that is correct.
So we're not talking about something that winds up in the protein or even something that is active as messenger RNA, but this must be the genome. The Toll-9 receptor must be an intracellular receptor, right?
It is. It's an intracellular receptor that binds to microbial DNA to induce an innate immune response.
So you could, they show this as fabricating, increasing the CpG content in the antigen itself, but heck, you could do this probably anywhere in the vector, right? And have the same effect?
Probably.
In theory, you could. And in fact, I had read it or misinterpreted it that's what they had done. Mm-hmm. But Stephen set me straight and said, no, they didn't want to mess with any of those other expressions, sequences, signals, and so forth. So they just change things within the antigen, the HA sequence.
Right, because if you change the other stuff, then you wouldn't know if your controls were working because you want to compare apples to apples.
And for the listeners out there who are all over this and right on top of it and are saying, but wait a minute, you're changing the code in something that's coding for a protein. How do you get the same protein? Well, you got to go back and look at your genetic code chart and realize that there's more than one codon for most amino acids so that you can make some changes without affecting the coding.
Right. And the idea of putting in more CpGs or removing CpGs is a pretty common thing when you're doing sort of thinking about immune responses and delivering DNA.
So, Brianne.
Yes.
Is this another Rube Goldberg thing? Why do we care specifically about CpGs? We being the immunology bureaucracy in the cell.
Because the frequency of CpGs is different in our DNA versus in prokaryotic DNA.
So we can distinguish between us and invaders.
Yes.
Okay. Great. Thank you.
Okay. So now actually with this experiment, they looked at T cell responses because they didn't really see any B cell humoral responses difference between the V3, V4 versus the V2, despite the fact that they got good protection from morbidity and mortality. So they looked at the T cell responses. And again, I could be a little bit on thin ice here, so Brianne can jump in. But basically, they found that the V3 and V4 that had the increased number of CpGs had good CD4 and CD8 responses in the lung and the spleen that were reactive to both the head of the hemagglutinin and the stalk of the hemagglutinin. And so therefore this CpG enrichment was maybe generating more antigen-specific T cell responses. That's what they said. I'm not sure I can explain that in any more detail.
That all makes a lot of sense.
Okay.
I could hand wave some reasons why that might be, but the data does not give me background on hand waving. Everything you said is what the data actually tells us.
Right. Okay. So what I haven't really said here, because I'm wondering if I skipped over it, but they do look at some of the breadth of the responses that they're getting with this, I just blanked on it, Cobra designed antigen. And so they are getting relatively broad responses in experiments that I haven't gone into, and I'm not going to, but we can if you want. But what they wanted to do then is just test, well, what if we just used a single hemagglutinin? And they chose to use the hemagglutinin from the California 09, from the challenge influenza that they've been using. So they use this non-optimized single hemagglutinin, and they put that into their same vector, and they call it V5. So now what I can't remember was, because it's just the hemagglutinin from 09, it's not going to have that CpG enhancement. So really what you want to compare it to is the V2, I think, that doesn't have the CpG enhancement. But in fact, they tested V1, 2, 3, or maybe 2, 3, 4, and 5.
2, 3, 4, and 5. I think.
Yeah, I wrote down V1, but it's 2, 3, 4, and 5 to look for this breadth of responses. And now, for those of you who are following along, we're already up to figure 7, and they see that there's a good breadth of responses to all these different influenzas they test: California 9, Michigan 15, Idaho 18, Hawaii 19, etc. And then they see that with V3 and V4, there are slightly broader responses than what they see with the V5 being the one with the single naturally occurring hemagglutinin. And the other thing that they found, I have it here in my notes, is at this point they didn't see liver toxicity or much liver toxicity, and they did this by measuring liver enzymes ALT and AST. And when I was reading it, I noticed that it was either in the figure, it was in figure 1 where they measured the ALT and AST, and I was like, they don't talk about that. But much later in the paper, at this point where we are now, is where they talk about the fact that these do not cause much liver toxicity. So that's a good thing. And they also looked at biodistribution, and I had fun asking about this because this is something that only shows up in the supplemental data, but they used bioluminescence assay on whole mice, and they were doing this intramuscular injection, and they see a whopping signal at the injection site, and they couldn't even assay or measure signal anywhere else because it was just so blown out by the injection site. So Kristen told me how she had to finagle the imaging of the mice so that wouldn't be the case, and she could actually see that there was some signal in the liver, but very little compared to the muscles. So basically, the virus was going where they put it and not trafficking too much to the liver. And then, as I discussed with Kristen and Stephen, Stacy's approach is, of course, let's do everything we can in mice because there's not nearly as expensive and precious as the ferrets are. So they have one pilot or one set of experiments, two different kinds of things that they did, where they inject ferrets. And ferrets are this really good animal model relative to humans for infecting with influenza. And they used just their V2 vector and infected ferrets, and then challenged them with the California 09, and then looked 9 weeks after vaccination for what kind of things they've been looking at all along. They looked for that neutralization endpoint titer, and it's really good with the V2 vaccinated mice and not with the naive mice. They look for protection from weight loss, and it's better when they get the V2. They
my favorite thing that they did, and the reason why ferrets are such a great model, is that ferrets allow us to model transmission of some of these respiratory viruses. And so they measured the viral titers in the ferret sneezes, because ferrets can sneeze and mice can't. So I was imagining little ferret sneezes when I was reading that part.
I wonder how you collect a ferret sneeze.
Probably with something like the Gesundheit.
Yeah,
oh, Alan, I can tell you, I met the Gesundheit machine guy.
Ah.
Really?
Yes. And I that's a story for another day. But also, Seema Lakdawala had this whole thing about the ferret playpen model for transmission, and showed movies of that in her talk at this meeting. So, I had forgotten this part about the sneeze, but in one of my exam questions in my class, I would always talk about the sneezate. Measuring the amount of virus in the sneezate.
Here, this is what it says in the methods, Rich. I don't think it will be as enlightening as you want it to be. Once anesthetized, PBS containing penicillin and streptomycin was dripped into their nostrils to trigger a sneeze response.
Oh.
The sneezes were captured in specimen collection cups.
Got it.
Well, they're knocked out, right?
Yeah.
Yes.
Okay. So you can direct this, and
you don't have to chase them around with an Eppendorf waiting for them to
or
sneeze.
A Kleenex or whatever. So
Yes.
and
I still kind of want to know how big the specimen collection cup is, though.
right.
Yeah.
And the sneeze is induced.
Yes.
Okay.
Yeah.
All right.
So basically they looked at the same kind of I don't want to say garden variety, but baseline experiments that they had done in the early part of the paper with the mice. And then they looked at the performance of this V2 in ferrets that had a previous history of influenza exposure. And making the story short, they saw that there was a good improvement in titers against all the strains. They tested California 9 and Memphis in their challenges compared to control injected ferrets. So this suggests that the ability of this adenovirus platform can increase strain-specific antibodies from past exposures, but also improve the breadth of responses to the new influenza strains that you might get exposed to. So that's all of the results. And then they spend a lot of time in the discussion, or not a lot of time, but a reasonable amount of time discussing some of the pros and cons of this system. And as we've kind of already alluded to, there is some prior immunity that humans have. Maybe about 30% of us will have some AAV immunity. And so being able to have these different capsid vectors for delivery might be an important thing. And that because of the gene therapy field, they're already ahead of us in thinking about all the ways that you might be able to circumvent that pre-existing immunity problem. The other thing that Rich pointed out is that because the thing is gutted and you can put in what you want, there's actually enough room in here to put both the hemagglutinin and the neuraminidase. So that's another nice advantage.
So one thing I want to know, and given that these vectors persist, did they look at persistence of antigen? And I'm worried that you don't want persistence of antigen for the life of the animal, right?
Or persistence of the viral DNA for the life of the animal.
Yeah.
Right.
But, you don't want to keep making antigen. So did they talk about that at all?
I don't recall seeing that.
No,
I did not
the persistence
that.
of either the genome or the antigen, I was on my mind the whole time, and I did not see any comment on that.
Yeah.
Right.
Yeah, a major reason AAV is used as a gene therapy vector is because the DNA that you put in with it sticks around.
Yep. Mm-hmm. Which may be why we don't have any AAV vaccines. Maybe it's a problem. I don't know.
Yeah.
There are some other AAV vaccines, maybe, not licensed for use in humans, but there are some that are farther along in this process than what's described in this.
There are, but the first one I ever learned about was from the Baltimore lab and involved actually expressing the antibody gene.
Yeah.
Instead of the antigen, because the idea is that you'd express the unique antibody for a really, really long time,
right,
and wouldn't have to make your B cells go through all the work of making it.
Right.
All right. I'm sorry, we have to move on, but thank you, Kathy.
Yes.
And
good. There's more papers coming out on this probably within the next year. So
great. All right. Our paper's from Nature Communications. It's called Viral Gene Drive Spread During Herpes Simplex Virus 1 Infection in Mice. And the first author is Marius Walter, Hake, Riley, Massa, Strongen, Klauser, Loprieno, Stenzlin, Santo, Roy Chuduri, Aubert, Taylor, Jerome, and the last author, Eric Verdon. These are from the Fred Hutch in Seattle, the University of Washington, and Montana State University.
And a place called the Buck Institute for Research on Aging in Novato, California,
Okay.
where if I'm not mistaken, the senior author is located there. I can make up stories about that, but Novato was close to where I grew up, and it was just a sleepy little town for a while, but it's in a wonderful area.
So I heard years ago at an ASV, Marius Walter, the first author, talk about this at its early stages. And so it's always been of interest to me, this idea of a gene drive. They're working on herpes because they want to understand how to perhaps interfere with latency, because you get infected by the herpes simplex viruses. It's for life. The genomes go latent and periodically reactivate. You make virus, you may get cold sores, you may get encephalitis, and we can't really do anything about it. And so they want to ask if we can use gene drive. It's a great name, right? Gene drive. What is it? It's some kind of genetic modification that makes a gene spread through a population, right? It's just exactly what it sounds like. And you can imagine it for all different systems. You can imagine it for mosquitoes, right? You could put a gene into mosquito and then it drives through the population. But they want to do it for herpes viruses and figure out if they could get a gene drive ultimately in latently infected mice. So they've already showed before for different herpes virus that it works. You can engineer a herpes genome, and what they do is they put CRISPR-Cas into the genome, and that will modify other genomes to put the sequence from that in it. They use fluorescent proteins just so that they can see them here, and at least in cells in culture, it works.
Yeah, so
Yeah.
the essence of a gene drive is it's a contagious genome or
Contagious
modification.
genome.
So
That's
that
right.
it will change, in this case, it'll change another genome, and then the original and that genome will go on and they'll tell two friends, and the whole thing will propagate until hopefully you've replaced the entire population of whatever you're driving the gene into with
Right.
things that carry that gene. And when it's done in mosquitoes, it's either done as an eradication strategy where you're sending some gene that is latently, it doesn't kill that mosquito, but it prevents it from reproducing maybe, or it renders it immune from being able to act as a vector for some virus, and so that spreads through the population.
So you can imagine a gene drive which attenuates a virus, right? And therefore it's no longer causing disease. Or you take an animal with a latent herpes infection, you introduce a virus that drives through the population of viruses in the animal and inactivates them in some way. So it's an attractive concept. But the key is you have to have co-infection of cells with different viruses, right? And one thing that blew me away here, gosh, they use a very low MOI and get co-infection of cells at very high rates. It's very interesting. But co-infection of cells happens a lot, apparently, at least with herpes viruses, as we'll see. And so what they want to know, as I said before, they've got this gene drive concept to work in cells in culture before. Now they want to know, can it work in an animal? And they use animal models of herpes virus infection. All right. And this is a proof of concept, as we said, for the first paper. They build a gene drive. I just love that you build a gene drive. It's like a transmission, building a gene drive that doesn't affect the fitness of the virus. So you're not looking at any
Engage the gene
fitness
drive,
effects.
Captain.
What's that?
Engage the gene drive, Captain.
Yes. Warp 1. Oh my gosh. So they put fluorescent proteins. They make the gene drive virus, the herpes virus. They put a fluorescent protein in and the CRISPR-Cas with the idea that this is going to transfer to another virus and push through the population. And there's, again, no fitness effect. So you don't have to worry about whether it's lowering fitness or not.
Well, so there's no fitness effect on the original virus, but doesn't it impact the fitness of the virus that it goes into?
Well, in the long term, yes. But in this case, it's a proof of concept where they just want to know if it'll work at all.
Okay.
Right?
And for a gene drive to work, you would need to balance, you need the driven gene to be able to survive and propagate in the population. So you can't do something that's like with the mosquitoes. You can't do something that's lethal because then you just drove to a dead end. So I think the idea with this would be like what Vincent said. You drive in a gene that attenuates the virus or maybe prevents it from reactivating.
Rich, you're muted.
Rich, so this is open access, and there's a really nice figure of this in Figure 1. So you might want to take a look at that.
Yes.
For now, the target for the gene drive in the recipient genome is the same as the origin.
Right.
So the targeted genome should not be any more crippled, which is not at all, relative to the origin. But as Vincent said, this is proof of concept. Down the road, you're going to want to design this so the end result is everybody's dead.
Yes.
Or everybody's stopped in their tracks.
Right.
Stopped.
Everybody's modified so as to no longer hurt you.
Yeah,
Yes.
Okay. Good enough.
So the cool thing about this is that, so they put fluorescent proteins of different kinds in the gene drive virus and also in their recipient, just as because they want to trace it. So you're going to get recombination using the CRISPR-Cas is going to give you 4 different fluorescent reporters, which you can see in a plaque assay.
Yes.
So you do plaque assay and you look at the color of the plaques and you can tell how the gene drive. I just think this is lovely.
And critically, this is going to give you a lot of really pretty pictures to print out and hang
Right.
out on your lab walls.
Right.
So the donor virus is one color, the recipient virus is another color, and then you can mix those up so you get 2 other colors. Crazy.
So yellow or cyan and cherry are the original wild type and gene drive viruses. So the wild type has yellow, the gene drive has cyan and red, right? And if you see cyan only or yellow and cherry, these are genomes that have been modified by the recombination.
One
So
other
it's-
nice thing about this paper is that they have this chart of what the viruses are. They have it in every single figure. You
Yeah.
don't have
Yes.
to keep going back
Yeah.
to figure one to look at
Yes.
to be reminded of which color is
Definitely
which.
appreciated that.
And one thing that I found a little bit weird was that the cyan M cherry plaque that they show, it should be purple, and it's kind of multicolored and reminded me of the rainbow
Rainbow,
paper
yeah.
that we had a long time ago. And they don't ever comment on that, and I just wondered if it bothered anybody else. Or just,
I
there's
see what
some-
you're talking about. It didn't bother me as
Yeah.
long as I
Oh.
can tell the difference between one and another. Now that
Yeah.
you pointed out, it does almost look like things are falling apart.
Yeah,
in
it's
the
not
middle of
100%.
It only
somehow.
bothered me because I liked the idea of seeing something be purple.
Mhm.
Yeah, I my assumption here was that what you're seeing is different. Since this is a plaque, you're seeing different stages of the life cycle.
Yeah. could
And
be.
since the markers are in different parts of the genome, I know herpes has this whole Baroque regulation system with its genes, and you may be looking at early versus late or that kind of thing.
Yeah.
So, first experiment, proof of this working. They use mouse neuronal cells in culture, and they co-infect them with their gene drive virus and a wild type virus, and they have some controls like the virus, the gene drive without the CRISPR-Cas9, right? And this is what killed me. They use an MOI of 1, where if you do the Poisson distribution, 25% of the cells are co-infected at an MOI of 1. That's pretty low. I mean, if I were going to design a co-infection, I'd do 5, MOI of 5, right? But it works. In fact, they can look at the colors of the viruses that come out, and they get anywhere gene drive viruses, that is made by recombination, anywhere from 20 to 80% of the cells are producing these. And so
The other
you don't
thing
see
is that at a multiplicity of 1, there's a lot of uninfected cells. So
Yes.
after you get one round, there's going to be more opportunities in culture
Yeah,
for getting
presumably.
co-infection.
Yeah, but it does work. It clearly
But
works.
I had the same reaction as you, Vincent. The minute they're talking about multiplicity of infection and co-infection, I'm going, ooh, Poisson distribution. I love this stuff.
Yeah. So basically, they show it works. And this is using 20% of the input as gene drive virus. And then if they make it 40%, the proportion of modified viruses, cherry expressing viruses basically, goes up to 95% after 3 days. So it's very efficient in these cells in culture, at least.
And as, conversely, or as a consequence, the wild type virus, that is the target of the gene drive, has almost disappeared
That's right.
in the 40% trial. So this is working
Yes, it's all been replaced
in
with
cell culture.
gene drive in cell culture. That's right. All right. So then they move into mice. They have a model for acute herpes infection in mice. The virus is inoculated intravitally, right? That is in the eye, infects the retina and other ocular tissues, and then gets to the central nervous system via the cranial nerves. And they talk about all the nerves. Most of you are not going to know what these nerves are. Different nerves transport the virus into the CNS. Before it reaches the brain stem, the virus infects the trigeminal ganglia, which are on either side, just below your ear. And that's in humans where the virus can be latent. And then it goes to the brainstem. So they inoculate mice with 1 million PFU in the left eye, and they collect the tissues at different time points, and they do plaque assays after 4 days to look at the color.
And because it goes by different nerves to the brain, it's going to go to different regions.
Yeah. And importantly, the different viruses that they're co-infecting reach similar titers. Okay, so it's not like one is outcompeting the other right away. So what they find is that in these experiments, they get about 15% production of gene drive virus, and then it increases. And it's interesting, it's different in different tissues. In the eye, it's 15 to 30%. In the trigeminal ganglia, it's 60%. And in the brain, it's 70%. So it works in the mouse model, but interestingly, it's different in different tissues. We don't know why that is, but you can imagine different things.
So to me, an important feature of this is that in contrast to cell culture, when you're shooting virus into a tissue in an animal, you don't know what the multiplicity is.
Yeah.
And you don't know how many co-infections you're going to get. And the mere fact that this works at all says you're getting co-infections.
Yeah, and I think that's one reason why it's important that they were using a low multiplicity earlier, because you don't know what the multiplicity is, but it's probably not going to be super high.
Now, for those of you who know this, DNA can break in the cell. And if it does, sometimes it can be joined by the cell to fix it, but that often results in problems. It's called non-homologous end joining. Many people study that. And that would make the virus resistant to gene drive. So they actually look at that to see if some fraction of the viruses, the wild-type viruses, are resistant. About 6% of the population seems to be messed up by this mechanism and is resistant to gene drive, which might explain why you never get 100% gene drive when you do these experiments. Then they try another infection model where you put virus in the foot pad of the hind leg. In this case, of course, the virus gets in the sciatic nerve and gets into the brain. And they, again, co-infect in the foot pad, and they look in different parts of the animals. The average proportion of gene drive viruses reach 60% in the brain, 50% in the spinal cord. So even by another route, it works. Co-infect, and where the viruses end up, gene drive has replaced a substantial fraction of the population. And again, they look very extensively at the different tissues. As Alan said, when it reaches the brain by different routes, it's in different places. And there's tissue-specific differences in gene drive propagation, the trigeminal ganglia, the brainstem, cortex, cerebellum, and so forth. You have different levels, which you have to take into consideration if you're going to use this therapeutically one day. They say in the brain it was highly heterogeneous with almost complete gene drive in some regions and barely any in regions right next to it. Yeah, the brain is a weird place.
Tell me about
Some brains more than others.
And they start to speculate about this, and they wonder if in some places it replicates the higher titer, and maybe that is what is driving the gene drive. But they found no correlation between viral titers and recombination. The eye, for example, they have the highest titers of virus in the eye, yet that's the lowest gene drive. So recombination doesn't correlate with viral titers, but they say maybe some other tissue-specific mechanisms. Now, let's see here. All right, I'm going to skip those experiments. They did a lot of experiments to look at infection by the different viruses in different parts of the brain and the eye and so forth. But so far, you've gotten the bottom line, which is that it works. But I want to move now to latent infection, right? So this is the key here. You want to be able to interfere with a latent infection. An animal gets infected, the virus reproduces, spreads to ganglia, and goes latent, periodically reactivated. Can you interfere with that with a gene drive virus?
So this is what we'd actually like to do clinically.
Yes.
Right. So before we were always infecting with the two types of virus at the same time. Now we're going to let the herpes virus establish and then use the gene drive virus to try to mess with it.
This would be the therapeutic approach. Now, unfortunately, they use a mouse model of latency, which isn't great, as you'll see. And so to establish a latent infection in mice, you put virus, you infect the eye by corneal scarification. So that's, you scrape the eye and then you drip virus onto it. And then you allow the virus to incubate, And then they will superinfect with their gene drive virus, let that establish, and then they induce the latent virus with a drug that turns on transcription, basically. And then the virus is going to be produced, the gene drive virus is there. And then they ask, can what we put in do gene drive on the reactivated virus, right? So they wait long enough such that the input virus that they began with is no longer there. It's all latent. They can tell by looking at infectivity. All right, and they do swabs of the eye to measure gene drive. So they do find gene drive working in this system. It's very spotty. Like they look at many mice, and only a few of the mice produce gene drive viruses. And in those mice, there's anywhere from 0 to 60% of the latent genome modified by gene drive. But it shows that it can work, right? Superinfecting virus can get in and do gene drive on the latent virus there.
Yeah, that's what I took away from this, and that is the superinfecting virus can access the sites of latency. And there is potential for transfer, but it's not robust.
Right.
Fair enough?
Yeah.
Mm-hmm.
And this, this system is an example of one of the many reasons it's so hard to work on herpes viruses.
Yes.
Because you've got this latent infection thing, and we don't have great simulations of it in models, and but so the fact that this actually works at all to any degree is really
Animal models are problematic generally.
Yes,
working with
and particularly in this case.
So one of the things that I realized in reading this paper is that maybe I'm not so good at reading. Because when I first read the title, and I thought about what I knew about gene drive, which was mostly in mosquitoes, I saw viral gene drive spread during herpes simplex virus infection in mice. And so I thought that there was somehow going to modify a mouse and it was going to spread its protection to its friends, its mouse friends, and get rid of all herpes virus in mice. And that was going to be amazing. And in fact, they're not thinking spread that way. They're thinking actually spread through the mouse from the infecting site, through cell to cell in the mouse from, say the foot pad or the eye or wherever into the mouse. And I think at the beginning I was like, oh, I was so excited at first. And as I thought more about it, as I read, it's amazing that they could do this much. And so, perhaps I had my poor reading skills gave me a very high bar at the beginning, but the idea that you can actually have this modification spread even among this organism and even have it partially work, but know that it's a possibility and something to work on more when it's so hard to do modifications in an actual organism with undisrupted tissues. It's very impressive.
I wonder if you relied on recombination. So I think CRISPR-Cas is the driver here, right?
Mm-hmm.
Yes.
Because if you make it and there's a target, it's gonna hit it, right? It's very efficient. But if you didn't have that, and their controls, they have control viruses without CRISPR, and they don't give you any gene drive. So the key is the CRISPR-Cas.
Yeah, and so to me, that's what I was thinking when I looked at the bits of data that says that the fraction of gene drive was different in different tissues, and it's different under these circumstances. It makes you wonder whether the CRISPR-Cas expression isn't sort of tissue-wise environmentally sensitive somehow,
Could be.
Or maybe the activity of the expressed CRISPR-Cas. Who knows?
There are other gene editing approaches now besides CRISPR-Cas, and maybe those are worth checking out, right? By the way, these mice that they use, Swiss Webster, it turns out that their eyes get scarred after the scarification and infection, and they're worried that that inhibits the uptake of their gene drive virus. So they use another mouse strain where this doesn't happen, and they get much higher levels of gene drive, like 9 out of 17 swabs were positive for gene drive viruses. And so that was a limiting factor in that mouse model. So basically, they show that both in cell culture and in animals, you can co-infect with a gene-drive virus. It will propagate through quite a fraction of the population, although not probably what you want. And this bears more research for sure, more tinkering. And they also say that there are better latency models. I think hamsters and guinea pigs.
Guinea pigs,
and
yeah.
they could try those and see what they get. But their idea is this kind of gene drive may be used in the future to limit the severity of disease. Now, an interesting thought experiment is, what would you do? You're not going to just put a fluorescent protein in, because that doesn't help. It's a proof of principle. But what would you do to the donor virus to attenuate a potentially lethal herpes simplex infection? You could deliver a deletion, I guess.
Right.
You could.
Yeah, I mean, I would think aim for the reactivation mechanism, like the regulatory system that reactivates the virus. If you can muck with that, your incoming virus, your modified virus, your gene drive would be able to do its active replication and go into the trigeminal ganglia and all that kind of thing.
Mm.
And along the way, whatever it co-infects that already has a herpes virus in it would get its reactivation nuked. And then none of these viruses would be able to reactivate. The one you put in would, have already been modified, and then it would drive its defective reactivation into the rest of the population. And so that patient would then still have a herpes virus in them for the rest of their life, but it would never reactivate.
Yeah, you could tinker with the transcriptional, like the immediate early promoter is needed for everything. Yeah. You could pull that out, for example.
Yeah, and the other thing that might work, totally speculation, but you mentioned, not getting the entire population of virus in the patient hit with this when you put it in. That may not matter, because during reactivation with a herpes virus, it reactivates and then gets transported retrograde through the neurons back out. And I'm thinking maybe if you designed this so that it would act kind of like a booby trap on the way out, like, okay, you reactivated and you're coming back down to make the sore, but then you encounter a neuron that has one of the gene drive viruses in it, and now, bang, stops the process. I don't know.
Imagine one day you have whatever herpes infection, you get injected with a virus and it takes care of it.
Then it's gone. Yeah.
Amazing, right?
That would be pretty
This
awesome.
is a long ways off.
Yes.
But
yes.
With
got to get into guinea pigs first.
So, Vincent, your idea of targeting an immediate early gene required for transcription, wouldn't that mess up future expression of the gene drive virus? Or would that not be a problem if it's a
right? It wouldn't be able to reinitiate its life cycle if you went with the immediate early genes that are required for it. But aren't there separate genes that are required just for reactivation or primarily for reactivation?
I think the reactivation idea is a good one. I mean, my first thought was, let's just go for the polymerase. But then I thought, but then you wouldn't be replicating.
Yeah, that's what I was thinking. Just rip the polymerase out. But the reactivation is largely done by cellular systems, right? Like histone deacetylases and so forth. So I'm thinking there's one main promoter in the genome, the immediate early, that's needed for the whole transcriptional program. But you're right, you can't screw up the gene drive virus. So, you can make the gene drive virus resistant to that modification so it doesn't lose the promoter.
Right.
But then I wonder if it would recombine with some wild type virus. And, so you got to test all this in cells and culture, right?
Yeah.
But it's and Marius now has his own lab, so he can do all this. That's cool.
Yeah. What I liked about this is that it's like a completely orthogonal approach to antivirals or vaccines. It's like just really different.
I mean, if you asked me if this would work, I said no way.
Yeah.
Because I would say co-infection is rare in vivo, but it's apparently not, at least for herpes in mice, right?
Yeah.
So it's very cool. I like it. Mm-hmm. I remember Marius because it was one of these afternoon talks. There weren't a lot of people in the room. And he came up to me afterwards because he recognized me. He said, "Oh, thanks for coming." I said, "It's very cool." And I said, "How do you get your fonts so nice on your slides?" And he told me his trick for doing that, which I use to this day. So thanks, Marius.
What is the trick?
So if you make in anything, if you use a different font, which I always do, and then it gets put on a projector that doesn't have that font, it's going to look all screwed up.
Yes.
Right? So he says when you're not sure where you're going to be projected, what he does is he makes all his slides and then he converts each one to an image, a JPEG.
Yeah.
And then he builds a PowerPoint with those,
Mhm.
and they will never change when they're projected because it's an image. It's not
Right.
pulling a font from the system. He told me that, and then I forgot it. So I emailed him months later. I said, "Well, how does that work?" So I saw this paper. I said, "Oh, that's Marius. Cool." But that was, a long time ago at an ASV. I don't even remember where it was.
So
I'm
the
glad to
Buck
see it's working.
Institute here, I've been flipping out over this because I wouldn't mind spending my career in Marin County. Okay. The senior author here, Eric Verdon, is in fact the president and CEO of the Buck Institute, which is over 20 years old and is dedicated to working on Alzheimer's. I notice here that the chief innovation officer is a gentleman by the name of Lee Hood.
Oh, I've heard of him.
And they got a bunch of people here with, chops. So, I used to fantasize with my fellow graduate students in a postdoc about the Drakes Bay Institute of Molecular Biology that we were going to establish so that we could go back and live in Marin in some place overlooking the coast, but it never happened. I went to Buffalo instead.
So, Rich, maybe you could apply and be the scientific communication officer.
Oh, there you go.
Yeah,
I could just-
so.
as long as I don't have to really work.
Right.
Okay.
Well, they might want you to leave Austin. So
Yeah, I'm
gonna
not
do
doing that. I'm not
Yeah,
doing that. I would,
You're too old.
I, if they want to move my daughter as well and her family, or maybe the entire Condit clan to Novato, I can do that.
There you go.
Pretty heavy price.
All right, let's do some picks.
Oh, Vincent, before you do, I wanted to tell you about because you were talking about Marius as the author, the two authors. One defended his thesis in August, Stephen, and Kristen is going to have her dissertation defense October 28th. So that's coming up really soon.
Cool.
So,
Yay, good luck.
yes.
Yep. All right, Brianne, what do you have for us?
I have a website that I learned about on social media recently. It's from something called the All of Us research program at NIH, which in and of itself seemed really interesting, but it is specifically a website about viruses and vaccines, meant to communicate viruses and vaccines to kind of a lay audience. And so as you scroll down, you can get a lot of different information about viruses with some nice images. And you can also see things like some facts about viruses are diverse. If you click there, you can actually see diverse capsid sizes and shapes of different viruses. Or if you click on how viruses work, you can kind of see the information about virus replication or things like that. So I thought that this was a nice kind of simple description of a lot of parts of virology, vaccines, things like that I could imagine students finding useful.
I
So
think this is really cool. Yeah.
This
As I
is cool.
scroll down,
Mhm.
it says a virus is a type of particle that can invade your body. Can you imagine how long they spent deciding to use the word
Deciding-
particle?
Yeah.
Yes,
Yeah.
how to define a virus.
Yeah. Because what are you going to say, right?
Right.
Yeah.
So interesting. Oh, is it a physics particle like on an atom? That's very interesting. By the way, this All of Us program is very interesting. I participate in that because they have a branch at Columbia. Basically, they take your vitals, they take blood, etc. And I don't know what they do with it, but it gets in a database, and They periodically contact you and tell you what they're doing, and they're trying to collect many, many, many people's, right, and make this huge database.
Yeah, as I was learning about it, I was learning about how one could get research access and sort of be involved in research using said data.
That's it, right.
This is really cool. Viruses travel light, it says.
Yes.
Mm-hmm.
Kathy, what do you have for us?
Doing this. Oh, sorry.
So I picked something that I heard referenced twice. So once in Australia by Anne Falsey in her talk. She's an RSV researcher at Rochester. And then last week we had Barney Graham here for a lecture, and he mentioned it. So it's an article in a publication called Undark, and the title is For a Pivotal Vaccine, Trial Error and Two Young Lives. And it was published a year ago in October of 2023 by Michael Schulzen. And it's about the two boys who died in 1967 from the RSV vaccine. And so it's a little bit of an untold story, and I think that's why both of these people mentioned it in their talks about RSV, because and it reminded me of Rebecca Skloot's book about Henrietta Lacks. So these two boys were Black. Their families had no idea until years later when Undark contacted them that their relatives had died from something that was this vaccine that went wrong. And so it's kind of a thing. And I don't have it here. I have it in something else that I didn't make it all the way in here. The two boys' names, so we should remember their names, Ross Hambrick and Victor King. And basically, for those who might not know, there was a lot 100 of an inactivated RSV vaccine that, in fact, a bunch of kids were vaccinated, 31 were vaccinated, and 20 of them, when they got infected, of those 20, 16 were hospitalized. So the virus infection of vaccinated children was worse than if they hadn't been vaccinated. And so it took many years to figure all that out. And it turns out, make a long story short, both a T-cell thing where instead of getting an interferon gamma type response, you get a dominant IL-4 response, which is the wrong thing you want for a virus infection. And also that the antibody response is to a post-fusion form of the RSV fusion protein instead of the pre-fusion form. And so that is a bad thing as well. At least that's my understanding of the two major ways that the vaccine was a problem. And so this is just a really nice, comprehensive thing. Like I said, kind of like Rebecca Skloot's book, but it's just a short article to understand a little bit more about what was actually going on at the time.
It's really great.
Important piece of journalism.
In the description of Undark that you put there, they say at the end, we're going to explore science as an occasionally troubling byproduct of human culture. It's interesting, isn't it? I mean, they say wondrous, contentious, occasionally, sometimes troubling. You think that's correct?
Yeah.
Sure.
It's sometimes troubling?
Yeah, we got to own up to what's been done and say, that's not the way this should be pursued.
Informed consent was completely different back then. And a lot of things changed subsequently.
Well, I think making this vaccine the way they did, that was troubling, right? It wasn't planned, but they screwed up, right?
And they had no idea. It took, like I said, it took years to kind of sort it all out.
But I agree, it's troubling in the fact that you have a bad outcome and you don't know why.
Yeah. But
that's how science sometimes is, right?
Yeah.
Rich, what do you have for us?
So Kathy gets an assist for this because of Kathy, the astronomy picture of the day is my home screen on my browser. Right? But this is more than about just the picture, the APOD picture that showed up yesterday. The picture itself is pretty cool. It's a picture of a rocket set to go off. That is a Falcon 9 on the gantry, bearing a space vehicle called the Europa Clipper, which I will elaborate on. It's taken at night, so that's all lit up. And in the background is the comet that's currently flying around and turning everybody on, okay? So that's a cool picture. But these APOD things have lots of links that you can click through. And ordinarily, were it not for how cool the picture is, I might have just gone to the links in particular about the Europa Clipper, because that's what grabbed my attention. So if you click through all the links on the Europa Clipper, you get to the NASA page and that kind of stuff. And this is a space vehicle that's been under development for some time, whose mission it is to go to Jupiter and orbit Jupiter multiple times, and in the course of orbiting Jupiter multiple times, have close flybys to the Jupiter moon Europa, which they believe has an underground sea that they think may be a salt sea, and so lots of water, and they want to know more about this. So that's the scientific mission. But I think what blows my mind more than anything else is the flight path that this thing is going to take, okay? So it blasts off, and it goes to Mars, and gets a gravity-assisted boost from Mars by looping around that slingshot effect, and comes back to Earth and gets another gravity-assisted boost. That takes 2 years, after which time it's got enough boost to take off to Jupiter, and it's going to take an additional 3 years to get to Jupiter, okay? And then orbit Jupiter. My God, this is Newtonian physics on steroids. You know, how can they plan all this stuff out? But it's very cool.
Yeah.
Talk about long-term experiments, huh?
Yeah.
Really, I'm just, my mind is boggled that you can, I mean, it shouldn't be, because it's all right there. It's just, you know, laws of physics and, you know, centuries of looking at the sky. But you can plot out a trajectory like that over a period of 5 years and have it work, and it'll work, you know.
But without the boost, it would just take longer to get there?
I think so. And what I couldn't find in all this that I would be interested in is before and after the boost, how fast are we going?
Yeah.
Right?
Yeah.
I don't know.
Yeah, I mean, the reason you use a gravitational assist is so you don't have to launch the whole mass of fuel that you would need to accelerate this thing to any reasonable speed toward Jupiter. And by slingshotting around a planet that is also orbiting the sun, you pick up its angular momentum, and then they're going to do it twice, once to Mars, once to Earth, and by then it's finally fast enough that it can actually get to Jupiter in like the lifetime of the researchers.
Some reasonable time before the investigators croak.
Yes. But while Jupiter still exists, yeah.
Cool. That's great.
Awesome. And
Alan,
I second the thanks to Kathy for APOD.
Yes.
Alan, what do you have for us?
I have a little, well, there's an article about it, and there's also a video you can watch. So this is some research going on at a deep sea hydrothermal vent. And these vents they're not a discrete spot. They're actually a pile of rocks on the ocean floor that's got, you know, hot water coming out of it. And what they wanted to do was dig under the rocks because, you know, on topside, when you turn a rock over, there's a bunch of cool life underneath it. And they wanted to know some stuff about these unique worms that you find, and are there larvae underneath the rocks? So they went down there with an ROV, and they're sitting on the ship trying to get the rock lifted up, and they couldn't do it. And the ROV pilot said, why don't we just take a crowbar down there? So they did. They sent the ROV down with a crowbar in its claw, and they crowbarred open one of the rocks, and it worked. And you can watch in the video where they're prying this thing up just like you would with a rock in the yard. And sure enough, it's full of all kinds of cool life, and there's a bunch of new science to do on that. But I thought it was cool, you know, sometimes the solution is just a crowbar.
Yeah.
In this video, these things, those white things, those are worms, aren't
Yeah.
they?
Yeah.
Oh, cool. Looks like somebody's garage.
Yeah, exactly.
Maybe yours. All right. My pick is a special issue of Cell called The Expanding World of Neuroscience. And it's full of articles on the different areas of neuroscience and where they are today and why they're really cool. Now, I clicked the download full issue, and it downloaded, and you get like 20 articles. And then it said, sorry, we can't, you have to pay to download this. But I already downloaded, so I don't get it. So someone should try it, because I thought people would like this, because it's a lot of nice articles about neuroscience.
Okay, my download will start shortly. Is what
Brain
it
body
says.
physiology, temperature, decoding the brain, a bunch of stuff about drugs, genomics.
We're unable to download all the articles unless you log in with a subscription. It's downloading something that's 114 megabytes. I don't know if that's all the...
I mean, I got 14 articles. It seems like that would be the content of an issue. They're all downloaded. And I got the same message. So
They know who you are, Vincent.
No, I don't think they know who I am. My home here, I don't have university access unless I enable it. So anyway.
And at the top, it says Cell supports open access.
Yeah, I noticed that too.
So anyway, try downloading it because the articles are interesting.
All right, I'm 50% downloaded. Maybe by the time we wrap up, I'll be able to give you a report on this.
All right, we have a couple of listener picks. One is from Jason. Hello, Twivers. Long-time listener, multiple pick sender, 1,000 epitope attendant, and big fan writing from Northeast Pennsylvania, not far from TWiV World Headquarters, where it's 11C with bright sunshine and a light breeze. Perfect fall weather for watching the fall foliage. I'll attach a photo of that below. Yeah, it was very nice. I didn't include it here, sorry. I looked at my drafts folder and found a couple of pics I'd stashed away, so here they are. First, a solar eclipse on Mars. It's not as dramatic because Phobos and Deimos are so much smaller than our moon, but it's still an awesome thing to see. And next, the Polaris program and their Harmony of Resilience, created with St. Jude's. It's an inspiring musical performance written by John Williams and performed all over the earth. And above it, I bet you can't watch only once. Keep up the great work. You inspire us too. And Ryan writes, he sends a link to an article in HealthBeat, New York. Ryan writes, this one is about the current state of vaccination rates specifically for New York State as of 2024. And the title is Childhood Vaccine Coverage Remains High, but we can't let our guard down. New Yorkers continue to be one of the state leaders in vaccine coverage.
This is from Marissa Donnelly, your local immunologist.
Right. Yeah, so interesting numbers. I have a graph here on the MMR immunization rates in different states. And yeah, New York is up there, Connecticut, West Virginia. Can you imagine West Virginia is high?
Not what I would have expected.
Oh my gosh.
Good job, West Virginia.
Yeah.
Why does New York have high rates? One reason is elimination of non-medical vaccine exemptions, which is the way it should be. All right, thanks for that, Ryan. That is TWiV 1161. You can find the show notes at microbe.tv/TWiV. You can send us your questions, your comments, your picks to twiv@microbe.tv. And again, if you enjoy these programs, please give us your financial support, microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University, Bio Prof. Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure. And I got 14 articles out of that download.
Yeah, but I think it works. I don't
think
know
that's
why they
the whole, yeah.
give you the message. So, yeah, I want people to be able to check it out. Good. Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Good to see you all.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.